The Old World monkeys are a group of primates which evolved in the same time and place as our human ancestors. An evolutionary tree describes the relationships between Old World monkey species. Biological anthropologists use molecular evidence, such as DNA, to estimate a tree. Assuming that the time at which two species diverged in the tree is related to the difference between molecular sequences, we can assign a length, proportional to time, to each branch in the tree. This is a recent evolutionary tree describing Old World monkeys. This group is interesting to study because the tree is well understood and there is a diverse fossil record for comparison to living species. Our main goal is to track evolutionary changes through time. Morphological data, the shape of the skull for instance, is one kind of data for which this type of analysis works well. We capture cranial surfaces with a laser range scanner. Using statistical techniques and visualization, we produce reconstructions of the hypothetical ancestors. The molecular tree for the living species provides a dated framework of relationships among major lineages, onto which cranial evolutionary change can be superimposed. In this first phase of our project, we selected 5 species representing distinct main branches of the Old World monkey tree. Surface scans of 5 crania from males of each species are shown at the endpoints of the tree. Crania at internal nodes are generated using our morphing method. The parameters controlling cranial shape are interpolated using the squared change parsimony method. At an internal node, a weight is assigned to each scanned cranium. The weight is approximately proportional to the inverse squared distance between the internal node and the endpoint. For each internal point in the tree, we want to create a surface representing the interpolated shape. Here is an overview of the morphing process we use. Let's focus on 2 of our input crania. We begin by carefully placing landmark points on the cranial surfaces to indicate how the 2 shapes correspond to each other. Next, we rotate and translate all of the input crania so that their different copies of their landmark points are as close together as possible. Then we find a weighted average target position for each landmark. Finally, we apply a nonlinear warp to each of the input crania so that its landmark points coincide with the target positions. The surfaces are now very close to each other, but not identical. The final step is to merge the 5 input meshes, producing a single surface representing the cranial shapes. Our interactive landmark editor is designed to make it easy for a user to identify homologous landmark points on the set of input surfaces. 2 individuals are shown at once to make it easier to see which points correspond. The user can also place curves or patches by manipulating landmark points on the surface. A user-specified number of points is then distributed across the curve or patch and projected onto the surface, establishing a detailed correspondence. Our sets of landmark points are fairly dense and represent both of the main surfaces of the skull, the vault, and the face fairly well. Landmarks are also more densely sampled in areas requiring more detail. This lets us warp the surfaces to be very close together before merging the surfaces. Once we have computed surfaces across the entire tree, we can run the evolutionary movie, so to speak, and watch how cranial shapes may have changed from the common ancestor to the modern forms. We know that our statistical averaging model of evolutionary change is not precisely correct, but it provides us with a testable hypothesis, including chronology, based on the data we have. The next step in our research is to compare some of the better preserved fossil crania to the reconstructed surfaces located at points in the tree where we think the fossils should fit based on their age and morphology. We could further constrain our estimates by actually including the fossil forms in the tree. We might even be able to improve both the estimated morphology and the placement of the fossils in the tree by iteratively removing one fossil at a time, recomputing the estimated morphology, and then reinserting the fossil. Note that our estimated shape is not 2-dimensional. It is a true 3-dimensional object, which can be compared in detail to fossil and modern crania. The 3-dimensional surface could also be used in other ways. For instance, we could create a real physical object for a direct study using rapid prototyping machines. Or we could analyze physical properties of the cranium and how they change during primate evolution.